Welcome everyone, and thank you for joining us today. My name is Luna, and I'm a technical application specialist at IVIN Technology. Today's presentation will focus on the latest advancements in live animal imaging at the cellular level. Our presenter, Hayden Kwon, Senior Technical Application Specialist at IVIN, will discuss the probability of visualizing immune response dynamics in live mouse model using intravital microscope. Hayden holds a master's degree in life science and has extensive experience in both clinical and preclinical imaging and research. During the webinar, if you have any questions, please feel free to use the Q&A box below. We will also address some questions submitted before the webinar. Hayden, you can start whenever you are ready.
Okay, thank you for the greetings, Luna. Hello, I'm Hayden Kwon, Application Specialist from IVIN Technology Technical Application Solution Team. So first of all, it is very honored to have you all today. So the presentation will proceed as follows. First, I will provide a brief overview of IVIN Technology's intravital microscope. Following that, I will present in vivo imaging application data performed by IVIN, obtained through visualization of immune cell dynamics in living mouse models. At the end of the webinar, you can see how manipulate the device for imaging in vivo lymph node with recorded video like a real virtual demonstration. Maybe you've been heard and aware of the conventional in vivo microscope, for example, the X-ray, PET, or MRI, which are macro-scale scope. So it was quite difficult to see the mechanisms of the disease development or drug biodistribution in live animals because you could only acquire whole body or whole organ imaging. So IVIN technology have been trying to develop and optimize the microscaled imaging system, IVM, for the cellular level visualization. And now we could observe various cellular dynamics, disease development, and drug efficacy and delivery inside of the living body on real time with sub-micron resolution. Utilizing this superb IVIM system, you could acquire the real-time 3D imaging in various cellular activities and molecular works directly, which allows for the direct visualization and analysis of delivery and efficacy of newly designed pharmaceutical compounds in preclinical studies. And I'd like to introduce our dual-mode confocal and two-photon microscope model in this slide. This is the most compact intravital microscope in the market, ideally suiting your researches. It has various key features such as cost and space efficiency, easy installation, and high usability, hands-free maintenance, and all-in-one package. So in here, I emphasize the dual mode utilization of confocal and two-photon laser. So in this imaging study, we have visualized the neuromuscular junction inside of Cy1YFP transgenic mouse, expressing the endogenous GFP signal in neuron. And blood vessel was labeled with anti-CD31 antibody conjugated with the fluorophore. So with this confocal laser, we could see the neuron and blood vessel with fluorescent signal, green and blue color each. But with two-photon laser, we could have seen the actin myosin structure with second harmonic generation signal without need another additional labeling step. If your research needs to see the accumulation of fiber, collagen, or other muscular structure, two-photon laser would be the perfect choice for your study. So IBIM is the world's leading company providing new all-in-one system with the best intravital imaging performance. All-in-one single box package enables easy installation, operation, and maintenance. And also it has co-optimized hardware and software for superb intravital imaging performance with ultra-fast polygon scanner and temperature monitoring system, motion compensation, and AI denoising software. So especially with automatic hassle-free motion compensation functions and various accessories such as live animal maintenance system and other various imaging window system for the wide range intravital applications, users easily can get the high quality in vivo data without any limitation in imaging various live organs. IBM's imaging system has ultra-fast polygon mirror scanner, speed up to 50 frames per second, enabling to capture the fast dynamics of cells like RBCs or the green immune cells flowing inside of the blood vessels in heart. With especially animal motion compensation function, it enables an automatic hassle-free, high-precision motion compensated imaging by accelerating the frame-by-frame motion compensation. It's the most optimal solution, particularly for the rapid moving organ like heart and the lung, as it can compensate the X, Y, and Z movement with the real-time acquisition of compensated imaging data right away. This is in vivo real-time real-time video of heart imaging. So in this slide, side-by-side comparison, even though raw imaging data has high degree of movement, but when we apply the motion compensation function, we can directly acquire the high quality of clear image can be obtained without any shifting artifacts. So without motion compensation, only this blurred image in the middle can be obtained due to the motion artifact, which disturbs the quantitative cellular analysis of the imaging data. So please note that any other conventional standalone microscopy only can acquire these shifted images from the same live animal model, which cannot even get the data for the in vivo analysis. So thereby, only IBM has optimized the in vivo solution, especially for only for intravital heart imaging in this cardiac research field. And to maximize the utility of the video rate imaging capability of IBM's microscope, we developed an AI model to efficiently denoise any real-time videos obtained by our microscope. The outcome preserved the high temporal resolution of the video while removing the noise. And surely during the noising procedure, even the small molecules like nanoparticle in red are also successfully imaged. It was very difficult before because previous denoising AI models lose temporal resolution with denoising procedure. So this work was published in Nature method last year, and IBM has an exclusive license for the global market. And also, we further developed a more advanced internally trained AI model to speed up the denoising processing more than 50 times. And I'd like to share the last key advantages from IBM technology. So especially for longitudinal imaging, we are providing in vivo imaging window chambers that are fully optimized for organ-specific applications. And we could successfully obtain the heart and the lung imaging data during using the specialized imaging chamber with tissue motion stabilizer to hold the tissue very still to reduce the artifacts induced from the heartbeats and the breathing. So in the case of imaging the liver, kidney, and other abdominal organs, we could implant this abdominal imaging window on the abdomen, which makes us acquire long-term imaging data, like up to almost 2 months. And thanks to the advantages of being able to image organs in a living animal's body at the cellular level, IVM Technology's current biomicroscope is used for imaging various disease models, including cancer models, dementia models, and sepsis models, including immune cells in lung and the lymph nodes. For now, I share the imaging data for obtaining the several immune. I introduced the technology of IVM imaging system. So from now on, I will share the imaging data for observing the wound healing and immune cell motility in living mice. So in this imaging study, we employed lysine GFP transgenic mice, which exhibit the endogenous GFP expression, specifically in neutrophils and macrophages. Additionally, we utilized an in vivo labeling kit consisting of anti-CD31 antibodies conjugated with the far-red fluorescent dye to stain blood vessels. So as shown in the video on the right, we could successfully see the blood vessels in red. And in the time-lapse movie captured at 30 seconds intervals, you can observe the active movement of immune cells. The image in the upper panel represents the ear skin of the control group, which was not exposed to the radiation, while the image in the lower panel shows the test group after radiation exposure. So given that both images are at the same scale, we observed approximately 6 times more neutrophils gathering at the radiation exposed site compared to the control group. Additionally, since the temperature difference between the non-exposed and exposed areas was only 0.2 degrees of Celsius, we further confirmed that this was an acute immune response triggered by radiation, not a result of temperature change. And again, most macrophages did not move around, so their motility changes are well correlated with the change in neutrophil and macrophage count before after irradiation. And in the second imaging experiment, we used CX3CR1 GFP transgenic mice, which express endogenous GFP in monocytes and macrophages to visualize the immune responses in ear skin. So a wound was induced with a 31 gauge needle puncture and KRS inhibitor known for disrupting cell-to-cell adhesion was applied 4 times. And imaging immediately after the wound, as well as 1 or 2 days later, we revealed a significant reduction in immune cell recruitment around the wound site in the KRS inhibitor-treated test group compared to the DPBS-treated control group. So this reduction was attributed to decreased cell-to-cell adhesion and migration in the test group. And here we could see the graph showing the quantitative analysis of monocytes gathered around the wound. So aligning to the previous imaging data with treating anti-inflammation drug, the KRS inhibitor, the immune dynamics has successfully decreased, and we could see as with our intravital microscope system. Next, I will share the result imaging data for tracking the immune cell motilities in mouse lymph nodes. Lymph node is small organ of immune system acting as filter or transfer the foreign particles. Lymph nodes are distributed the whole body and connected by lymphatic vessels. Although lymph nodes are far away from each other, they function as a single entity mainly because of its principal cellular constituents, the lymphocytes. The lymphocytes are intrinsically mobile and continuously recirculate in large numbers between lymph nodes via the blood and the lymph. This data showcases the various immune cell movements within the popliteal lymph node. Additionally, in the previous slide, we observed in this slide we could see the B and the T cell movements in the lymph node. And similar to T cells, B cells were also visualized passing through the fibroblastic reticular cell (FRC) network of the lymph node and exiting the outward. And in the lymph nodes, there are specialized blood vessels called high endothelial venule, or briefly called HEV. So you can easily find HEV in lymph node because of their large size and the thick vessel wall. HEV is a main entrance into the lymph node for circulating lymphocytes. So in normal condition, HEV allows only lymphocytes to enter from the blood. For the last decade, multiple steps of T lymphocyte entrance in HEV and related oxygen molecules have been identified. However, the exact behavior of interaction between rapidly flowing T lymphocytes and endothelial cells remains to be analyzed because of the lack of proper imaging system. So, in addition, the transmigration of T lymphocytes across endothelial cells has not been clear because of the proper imaging method as well. However, with our IVN technology's real-time in vivo imaging capability, we were able to observe the migration of cells passing through the HEV. We stained the FRC, the fibroblastic reticular cells, which compose the microarchitecture of lymph node using elixir conjugated anti-ERCH7 antibody displayed in blue. And additionally, we injected an in vivo labeling kit composed of CD31 antibody to stain blood vessels and CD3 antibody to stain T cells, T lymphocytes, resulting in red and green signals for each cell type, respectively, before proceeding with imaging. So in this video on the right, you can observe the T cells, green T cells, flowing in the lumen of vascular endothelial cells, and then adhering to the vascular structure wall, and subsequently migrating towards the FRC side, which is the outer side. As you can see in the video, we were able to visualize in real-time process where T cells, T lymphocytes flowing within the blood vessels in the lymph nodes adhere to the vessel wall, pass through, emerge, and all within just a few minutes. And this data reveals that not only T cells but also B cells utilize the perivascular channels within the lymph node to traverse from the HEV lumen to the blood vessel, and even though the fibroblastic reticular cell network. Remarkably, we found hotspots for the trans-EC and trans-FRC migration of T and B lymphocytes. Interestingly, T and B lymphocytes preferentially shared the trans-FRC migration hotspots but not trans-EC migration hotspots. Furthermore, trans-FRC migration of T and B cells previously occurred at FRCs covered by CD11C positive dendritic cells in HEV. So these results suggest that HEV endothelial cells and fibroblastic reticular cells with perivascular dendritic cells delicately regulate T and B lymphocyte entry into the peripheral lymph node. So with iBeam's intravital microscope, you could get high quality live cell imaging in almost all internal organs, including brain, intestine, and the prostate, as well as ear, skin, and the lymph node in live mouse models. So far, I've shown you the imaging data that we performed before. So now let's move on to the virtual demonstration session to show you the actual imaging process for lymph node visualization. So this is the brief scheme for today's lymph node imaging. So we anesthetize C57 B6 wild-type mouse and then remove the hair on the right side of the body. So after exposing the lymph node by simple surgery, we conducted lymph node imaging using anti-CD31 antibody conjugated with the specific fluorophore to stain the blood vessels in red, and also the green nanoparticle injected to see the blood flow. So I started the video. So I will explain the real procedure of the imaging process. So at the top, you can see the main body of the microscope, or at the bottom, there are various supplies. So we'll notice three main devices: the main power supply, microscope, and each power supplies and controllers for the two photon and the confocal laser. Next, I'll explain the main body in more detail. So there is a stage where the animal model is placed for the imaging, and on the ceiling there is a turret that can hold up to 5 OP lenses, and next to it there are ports where you can connect up to 5 temperature sensors. So for example, if you want to keep the stage warm to maintain the mouse's body temperature, you just need to connect this cable to the port. Once connected, the animal maintenance system automatically measures the temperature and adjusts it to the set temperature to maintain the mouse's body temperature. So you can also use the rectal probe to measure the core body temperature of the mouse with just one click and adjust it as necessary. And using the jog dial at the top, you can move the stage along the X, Y, and Z axis to proceed with imaging. And I will show you the operating mouse in greater detail. So today I prepared for imaging the right inguinal lymph node of the mouse by anesthetizing it, shading the area, and then making an incision to expose the lymph node. Afterward, I placed the cover glass over the tissue to prevent it from drying and to match the refractive index required by the lens. Now I will insert the stage into the chamber, the imaging room, close the door, and begin imaging. So, okay, here we have the IVM engine software featuring a user-friendly interface. So on the main screen, you will notice the separated displays for the DAPI, GFP, RFP, and the Sci-5 channels. And additionally, there are displays for the integrated live video, main display, and window for the captured images. And to ensure the optimal excitation and the detection during in vivo imaging, I've already adjusted the laser power and the detector value. So by activating the scanner and the shutter, we can locate the focal plane until the signals are displayed, enabling us to observe the morphology of the lymph node blood vessels. So using the jog dial, we can navigate to find the best field of view on the cover glass. So you can see the large blood vessels with multiple branches and vasculature as their ideal areas to capture the signals. So in this experiment, the red signals represent blood vessels labeled with anti-CD31 antibody conjugated with the fluorophore from the Sci-5 channel. And the green signals represent the fluorescent particles. So we can observe the green labeled particles in action as they flow inside of the endothelial surface or firmly adhere to the blood vessel with some sense of purpose. So with each observation, we inch closer to unlocking the secret of vascular endothelial dynamics and the delicate balance between drug response and circulatory function. So for this imaging session, I will be using the C-stick imaging mode. So this approach allows us to obtain a comprehensive view of the vasculature across its entire depth. So find the right focal plane, and then we will load the Z coordinates to capture the images, starting from the top to the bottom with this load position tab, and inserting the 10 micron interval. So the imaging depth will be 60 microns. Once the images are captured, we can use the IBM Studio, the post-processing software, to stack them together. And also you can use all the functions in combination, but to be respectful of your time, I use both the stack and the mosaic function at the same time. So move the stage to find the suitable focal plane and turn on to get a large field of view imaging data of the target organ. We could utilize this function. So just load the start and the final position with this load key. So for now, we will get 2 by 2 total 4 images, and stage will move automatically after starting the imaging session. So during imaging, once you've done the setting, there's nothing to do. So during the imaging session, I like to share the imaging data that was captured before this imaging. I use the same eye, so you can see the same vasculature in this slide. So on the left side of the screen, you can see the lymph node imaging taken with the 25x lens, providing the high resolution view. And at this spot, I recorded the video so I could get the information about the movements of the particles in the live mouse. And also, on the next slide, yes, there is a video extracted from the 3D build data, allowing us to visualize the vasculature of the lymph node from the different angles. And also, I extracted the orthogonal video from the surface to the bottom to look into the vessels and particles more precisely. Okay, so let's move on to demonstrate how to post-process the acquired data using the IBM Studio, our software for 2D, 3D, and 4D in vivo data processing. So please note that due to the time constraints, we may not be able to cover every function in this short demo. So by adding the Hive directory folder into IBM Studio, you will have access to all the categorized files. So within the software, you can adjust and improve the quality and visibility of the images, such as modifying the color balance and the brightness, the contrast of each individual image. So with adjusting the color balance part, you can see the balance is changed during this processing. So like this, once the necessary adjustments have been made, we can play a set of C-stick images with the C-stick stem and the scale bar inserted for the better visualization and analysis. So this allows you to observe the acquired images in a sequential manner, gaining insights into the lymph node structure across different focal planes. And it is completely possible to perform basic functions such as making reproduction, and furthermore, quite time-consuming and demanding one like making mosaic, also called stitching. And additionally, we can utilize the depth-wise acquired datasets for 3D reconstruction. So by making a few adjustments within the software, we can generate a video clip that provides a horizontally rotating 3D view of the lymph node structure. So this visualization method complements the 2D images and offers a comprehensive understanding of the tissue's organization and the architecture. Throughout this webinar, we have explored the process of in vivo lymph node imaging using the intravital microscope along with the associated hardware and the software. So thank you for your attention, and if you have any questions, please feel free to ask through the Q&A box.
Okay, so thank you very much for a great presentation. We will now take some time for questions. So first question is, how many days can I observe the target cells in living body with imaging window?
Okay, so we are providing the several windows. So when using an imaging window for with this intravital microscope, the duration of tissue observation depends on the several factors, including like window design, tissue health, and imaging setup. And with IBM's intravital microscope, most tissues could be successfully imaged for over a month after window implantation without significantly affecting tissue viability. And for example, in the case of the cranial imaging window model in mice, the brain could be visualized repeatedly over weeks, months, or even up to 1.5 years. So it is very good animal proof for studying the various disease models like Alzheimer's or other neurodegenerative diseases. And we offer support with online or on-site training for imaging any target organ or tissue using our system.
Okay, thank you. And for the second question is about the imaging tab. So for example, if we take the imaging and without hamper the skin, or for example, if we want to do the brain imaging, if we need to cut open the skull. So please, Hayden, can you answer the questions? Thank you.
Yes, for visualizing the cortex on the brain, we need to cut and open the scalp, and also we made a 3 millimeter diameter hole on the skull. So for getting a better quality of the visualization, we implant the 3 millimeter diameter cover slip and attach it to the scalp and the skull with a specific dental cement. So it makes this window stay about over a year.
Okay, so yeah, I think there are so many questions about how to do the imaging and how we expose the organ and how to take it on the lens. So I think Hayden has already told us how to do this. So it looks like we have already covered all the base. The webinar will be able on demand on our YouTube channel. So at IVN Technology, we offer both hands-on and virtual demonstrations, along with free imaging test experiments using your samples and the training for animal surgery models. So for any question, feel free to reach out to us at information@ivntech.com. Thank you once again for joining us today, and we look forward to seeing you next time. Thank you.